Hey everyone, let's talk about blood today. So we're going to do kind of just like an intro into blood, and a much deeper analysis will happen probably later on in AP2 and definitely into our professional programs. But if you can recall, we've talked about blood a few times throughout the semester, but blood is technically a type of connective tissue. And if we think about connective tissue, this is one way that you could understand connective tissue is that it connects two things together, like our ligaments connect two bones together, or our tendons connect a muscle to a bone. Pretty standard connections. Well, what does blood connect? Blood kind of connects all of the tissue of the body with one another. So like if the brain needs to communicate with the GI tract, it can drop a hormone in blood and allow that blood to send the signal to that effector organ. If we need to transport oxygen from the lungs to the different tissues of the body, we can grab onto some oxygen with some blood coming near the lungs, and then that oxygen can get transported around the body. So in that way, oddly enough, blood is a type of connective tissue. What kind of connective tissue is it? It's a fluid connective tissue. And it's a fluid connective tissue, meaning that it has some liquid elements and it has some stuff floating around and dissolved in the liquid, and then some stuff not dissolved. It's just floating around in the blood. And so let's continue with that. Let's talk about what is blood made up of. Well, blood, if you take blood and maybe take a vial of it and let it sit there, it'll eventually separate out into its solid components and its liquid components. Or we could test this really fast with like a machine, but you can also just put it into a centrifuge and spin it really fast, and that's also going to separate out these solid and liquid components of blood. And given that we have stuff floating around in blood, but it will separate out after a while, we would say that it's a heterogeneous mixture, right? As opposed to like a homogeneous mixture where something is dissolved in water, and if you let it sit there, it's just not going to separate out at all. Blood is a heterogeneous mixture, meaning that it will eventually separate out if you give it enough time, or if you spin it really fast and separate it out. So what can we separate blood out into? If we put blood into a centrifuge, what are its components? Well, at the top of the vial after we spin it will be all the liquid stuff, and this is what we call plasma. It's mostly water, but it's got some proteins in it. It's got some ions dissolved in it. There might be some nutrients like sugar and stuff in it. And there's certainly going to be hormones, like we talked about from the endocrine system, floating around in this plasma and using this liquid base as a way to dissolve and float around. And then we've got the solid components that would then sit at the bottom of the vial. And for the most part, we're going to be talking about red blood cells, or erythrocytes, and that makes up the majority of the solid components. However, there are a couple other solid things in there. There are white blood cells, and then there are these things called platelets, which are not technically cells. I'm going to talk about them in a little bit, but they're like little fragments of cells. But those major pieces create what is called the formed elements. We can't really call them cells because platelets are not cells, but you can sort of think about them as being the solid cells that float around in the plasma. And again, those are red blood cells, white blood cells, and platelets. Those make up the formed elements. And for the most part, the ratio of formed elements to plasma is about 45% to 55%, 45% being the formed elements, the solid stuff, and then 55% being the liquid parts. And given that we have these different elements or these different parts of blood, what does blood allow us to do in the body? What are the major functions of blood? Well, first and foremost, we can transport stuff around the body. We can transport stuff like oxygen, carbon dioxide, nutrients from the GI tract, and then spread them out around the body. Hormones from the endocrine glands that we've talked about, and even the waste that our cells produce that we need to get to the kidneys and the liver to eliminate the waste. So we can transport stuff around the body, given that we have this liquid, fluid, connective tissue floating around through the body. But there's a couple other important functions of blood besides just transportation. One important function of blood is its ability to regulate stuff around the body. And we've talked about one of the regulation capacities of blood already, and that's its ability to regulate pH. So we need to have a middle ground pH in our blood in order for the cells of our body to function properly. If our pH gets too high, too basic, our cells really don't like that, and it's hard for our cells to function properly. If our pH drops too low and it gets too acidic, it's also difficult for us, our cells, to function properly. So we have the way of buffering or keeping our blood within a normal pH, a normal blood pH homeostasis. So that's one ability for our blood, is to regulate our pH for the cells of our body. In addition to that, we can regulate the amount of fluid that is in between and around and sometimes inside our cells. So our blood, we can balance the amount of fluid in our blood that will help us balance the fluid that is around and inside of our cells. If we notice that there's too much fluid in between our cells, we might pull off some of the fluid in our blood and help pull some of that fluid off of the cells and off of the interstitial fluid in between the cells. Obviously, in another sense, if we were dehydrated and that fluid level is dropping in our cells and in between our cells, we can add more fluid to the blood and help balance that fluid content. And then finally, one that's kind of interesting is we can use blood to help us regulate our body temperature. Because our body mostly produces body heat in our big skeletal muscles, it's kind of asymmetrically distributed around the body. The heat is, that is. So the heat gets made in our muscles, but how do we get that heat to the other tissue, like our nervous tissue in our brain, or the organs in and around our abdominal cavity? There's not a ton of skeletal muscles in between and around those, so how do we get the heat there? Well, we can pump blood through the skeletal muscles, and it will grab onto, or it'll warm up, it'll grab onto the heat, or it'll increase in temperature, and then as the blood moves around the body, it can distribute the heat nicely around the body, and we can adjust the amount of blood that flows close to the surface of the body. In that way, we can regulate how much heat we're radiating off, and that helps us regulate body temperature as well. So we got transportation functions, we've got regulation functions, and then finally, the last major function of blood is a protective function. It helps protect the body in several different ways. I'm going to ask us about two of them. I'd like us to know that we have white blood cells in our blood that help us with immune defense. So it fights off pathogens, it removes debris that we don't want to be in our body. So there's an immune aspect to protective aspect to our blood. We'll continue to talk about this more in A&P2, but I'd like us to kind of be aware of it right now. And then finally, we can use the clotting ability in our blood to protect us against excessive blood loss. So if a blood vessel bursts open or an organ gets ruptured, our blood can target that area and clot and prevent blood loss from that site of injury. And we'll talk about that more in detail as we keep going here. So again, I'd like us to know the major functions of blood being like transportation, regulation, and protection, and then some of the examples that we just covered. Okay, let's now talk about what tests are commonly run on blood. And if you're already working in healthcare field, you probably know of a bunch of different tests that we run on blood. I think there's now like a kind of a complete blood panel that would run all of these tests and more, but let's just talk about a few of them. First up, we can test what's called hematocrit. Hematocrit, or HCT. And what this does is it measures the proportion of the solid parts of the blood and the liquid parts of the blood. And that's useful for us because, again, we want to kind of see like a 45% to 55% split between the formed elements and the plasma. And this is useful information to medical professionals because it helps us assess the oxygen carrying capacity of the blood that we just took out. So if somebody's feeling really fatigued and they're super tired and they come into our office, or they're on our floor of a hospital, we might just quickly run a hematocrit test just to see if they have really low red blood cells. And we're going to maybe assume that if they do have low red blood cells, maybe their oxygen carrying capacity is pretty low in this blood. So that's one useful part of this test. But what if we take someone's hematocrit and we notice that they have way more red blood cell to plasma than they should? So instead of being like 45%, what if it was like 55% red blood cells or even 60% red blood cells? What might we assume there? Well, if we notice that there is not enough liquid in someone's blood, then we might assume that that they might be dehydrated, that their cells have pulled off too much of the fluid in the blood, and the downside to that is that red blood cells, without enough fluid kind of bathing them, they get really difficult to pump around the body. So it's not ideal to have too high or too low red blood cells in our blood, and we can test that easily with a test of hematocrit. Couple other tests that we can chat about, and these are going to make more sense as we start talking about the different elements or the different parts of blood. But we can test for hemoglobin, and the purpose of this test is to measure the amount of hemoglobin, which is a protein in the red blood cells. And so although maybe sometimes the red blood cells themselves are in the correct number, if someone has a really low hemoglobin levels, which is the protein inside of the red blood cells, it still might limit the oxygen carrying capacity of that individual. And so maybe if somebody has a really low iron count, it might not, the body might not make enough hemoglobin, which is crucial for oxygen carrying capacity. We can take a differential white blood cell count, and this kind of measures the amount and then percentage of each of the white blood cells or leukocytes. And this can be an easy way for us to test if there's an immune response in the individual already taking place, or if the white blood cells are really depressed, it might cue us into that this person's immune system is kind of depressed and maybe is susceptible to diseases or infections or something like that. And then finally, we can take a platelet count, and a platelet count measures the number of platelets in the blood. And we just said that platelets are useful in blood clotting, so it's nice to know how well the blood can clot. If we're about to do surgery, we might need to consider how well this person's blood can clot or not clot. Maybe if the blood has a really high platelet count and a high ability to clot, we might also think about giving this person like blood thinners to prevent over and excessive and unwanted blood clotting. So kind of the major tests that I'd like us to be aware of are things like hematocrit, especially, and then things like hemoglobin tests, differential white blood cell count, and then a platelet count. Just some basic blood tests for us to cover. So let's next talk about each of the components of the formed elements of blood. We'll talk about red blood cells, white blood cells, and then platelets. What are they? What do they do? And what do we need to know about them in terms of anatomy and physiology? So first off, let's talk about red blood cells. Sometimes those are called RBCs. And the main way that we refer to red blood cells is that they're called erythrocytes. So you hear them kind of called any of those three: red blood cells, RBCs, or erythrocytes. So a red blood cell is kind of interesting because it is one of the cells of the body that does not have a nucleus inside of it. When red blood cells are created, they come from mitosis, and they're sort of splitting and dividing and creating a bunch more cells, and that happens in the red bone marrow. And when a red blood cell is maturing and about to leave the red bone marrow and start flowing around the body, the nucleus that's inside of it gets ejected out of it, and it gets recycled. And you'll notice that the big round cell, when the nucleus gets ejected, the center of it kind of deflates a little bit so that it looks like a kind of an inner tube with the center part still there, or maybe like a jelly donut that has the jelly sucked out of the center of it. We'll see these two depressions on the top and the bottom of the red blood cell, and we call this having a biconcave shape. And this shape is necessary for red blood cells to properly grab onto oxygen and carbon dioxide. So the main idea here is that when red blood cells are created, they eject their nucleus, they create this biconcave shape, and by not having a nucleus, they create more space for the really important protein on the inside of it called hemoglobin. And what hemoglobin does is it can grab onto those two gases that we just mentioned, oxygen and carbon dioxide, and that's what makes a red blood cell really good at performing its main function of gas exchange. So we can pump these red blood cells to the lungs, we can grab onto some oxygen, pump that oxygen around the body, and any cells that need that oxygen can grab onto it. They can undergo cellular respiration, and then when the cells are done using up the oxygen, it's converted into carbon dioxide, which is a waste product. The cell doesn't need the carbon dioxide anymore. And the red blood cells then can grab onto the carbon dioxide and then float back to the lungs, dump off the carbon dioxide, grab onto the oxygen, and that whole process starts over again. But if you think about it, a red blood cell not having a nucleus means that it's not very good at creating more and more proteins. Remember, the nucleus is where the DNA is held and where the site of most of the ribosomes are created that make proteins. So it's not very good at creating new proteins, which means that erythrocytes have a start and an end to their life and need to be recreated constantly in the body. The normal erythrocyte lasts for about 120 days. We're talking like 3-ish months that this red blood cell can float around the body and help perform that gas exchange. And after that life cycle, the proteins inside of the red blood cell, they start to break down. The red blood cell itself starts to break down. And we have two major organs that can notice these broken down red blood cells and recycle them. And most of that happens in the spleen, and then a little bit of it happens in the liver. And those two organs, their main job is to break down the red blood cell, recycle the parts so that we can reuse them again. And the main protein that's inside of the red blood cell, we said, is hemoglobin. And our goal here is after the normal life cycle of the red blood cell, we should recycle most of the parts of the hemoglobin. So I would like us to know the three major parts of hemoglobin and what happens when we recycle red blood cells, what happens to each one of those parts. So let's go over those next. Hemoglobin is formed by three major parts. There's a heme group. There's an iron molecule at the center of that heme group. And then there's 4 huge proteins called globin. So hemoglobin has a heme, a globin, and an iron component to it. And when these red blood cells are being recycled, we really want to recycle each one of those parts of the hemoglobin so that they can be either reused or sent off and excreted while we make more red blood cells. So let's go over each part. Heme is broken down into a protein called bilirubin. So after a red blood cell is kind of nearing the end of its life cycle, it starts to break down. The heme turns into a protein called bilirubin. And bilirubin is the classic yellowish, orange-ish molecule that we can notice if a person has a buildup of bilirubin in their system. We can sometimes see that orange or yellowish tint straight through their skin or in the white parts of their eyes. And so that's a really useful clue for us that maybe the liver is not breaking down that protein effectively enough. If we notice that there's a bunch of bilirubin in someone's system, we can probably assume that they might have maybe a liver issue there. And so normally what happens is when bilirubin starts to accumulate, because heme is getting broken down, the liver then breaks down bilirubin, and then it eventually just gets excreted in our urine and in our feces. And this broken down bilirubin is what gives urine kind of the yellowish tint and what gives feces its brown color. Next up, the globin parts of hemoglobin, these are these 4 huge proteins that make up hemoglobin, and the globin parts are broken down into their individual building blocks of that protein. And the individual building blocks of protein we know are amino acids. So globin gets broken down into amino acids, and they get recycled to make new proteins. Some of them just get recycled straight back to making new red blood cells, and other ones get recycled and used elsewhere. And then finally, the iron that's at the center of the heme group is kind of interesting. Iron's kind of hard for our body to get a hold of. It's hard to absorb iron from the food that we eat. We're not really, really good at doing that. And we really want to save the iron from each one of these red blood cells. We don't want to excrete it off. We don't want to break it down. Instead, we want to save it and reuse it for future red blood cells. And so the iron part of the hemoglobin gets broken down and it gets stored in the liver, and then hopefully it will get reused in red blood cells, because it's kind of hard to get a hold of. If you have a low iron content, it might be difficult to make these erythrocytes and the hemoglobin that's inside of it, and it can result in sort of increased fatigue. You might feel tired all the time. It could be decreased iron levels. Okay, so that's red blood cells. Let's next move on to leukocytes, or white blood cells. So leukocytes, white blood cells, sometimes are called WBCs. We have two major categories of white blood cells. They're the granulocytes and the agranulocytes, meaning if they have a granular-like pattern to the surface of the cell or not granular-like pattern. And I'm not really going to ask us to be able to identify the difference between these cells just by looking at them, but I have some slides in our notes that can help us do that. I think in other classes, though, they really do like us to be able to just look at these cells and be able to identify which one is which. So let's go through the main white blood cells of our blood. First up, we have neutrophils, which are one of the granulocytes, because it has a fill at the end. Our neutrophils, these are the most abundant white blood cells. They make up the majority of our white blood cells, and their job is to respond to infection or injury right when it happens. So these are like our first responders. As soon as we notice that there's a foreign body entering our blood, or there's a cut in a blood vessel, these neutrophils are going to fly to that area, and they're going to try to phagocytize, or they're going to engulf and then eat and break down stuff like bacteria, cellular debris, anything that's getting into our blood that we really don't want in there. And these, again, are the neutrophils. These are basically first responders of the white blood cells. Next up, we have eosinophils, another granulocyte. Eosinophils are probably one of my favorites. Eosinophils are really good at combating parasites. So they're going to be floating around through the blood and looking for parasites that have sneakily entered the blood and are trying to evade any of the white blood cells and trying to live in our blood. And the main one that these ones look for are these tiny little worms called helminths. It's not really like the tapeworms that we would find in the GI tract, which are gigantic. These are like tiny, tiny, tiny little blood-borne worms. And when eosinophils see them, they get targeted to that area and they try and engulf and they kind of like attach to it and start digesting the worm from the outside. In addition to just combating these parasites, eosinophils are involved in allergic reactions, and specifically what they do is they break down histamines, which are, we're going to talk about these in a second, but they break down histamines which cause inflammation. So they kind of come in at the tail end of maybe an allergic reaction or an asthma attack, or any time that a histamine is released. And after that allergic reaction has taken place, the eosinophils come in and break down the histamine and slow down and reduce the inflammation in that area. Speaking of inflammation, let's talk about basophils next. This is our last granulocyte. There's only a small amount of basophils in the blood, but they have kind of an important role. These ones play a major role in allergic reactions and hypersensitivity reactions. So if you or you know someone who's allergic to something like peanut butter, or maybe pollen, or a bee sting or something, these cells are what are producing that allergic response. And what they do is they look for specific foreign bodies that they think should not be inside the body, and they produce an allergic response by releasing histamines. So when they notice that there's a pollen in our bloodstream, or that we've just inhaled, they're going to be targeted to that area. They're going to cause some inflammation. That's going to increase blood flow to the area. It's going to dilate the blood vessels. It's going to allow a bunch of white blood cells to hit that area in case this foreign body that we noticed needs an immune cell response. And occasionally what happens is, with really severe allergic reactions, you'll see that the blood vessels will really dilate and tons of blood will flow to that area. So we're going to see like a swelling in that area. increased blood flow, increased heat. Occasionally, by dilating the blood vessels so much, it allows a bunch of water, a bunch of the plasma to exit the blood and fill the tissue in that area. So you might see a bunch of bumps on the skin in that area. It might turn into to hives, so like a really severe swelling reaction. It might even constrict the breathing tubes, the bronchi, and cause like an allergic reaction where we'll notice that the individual might not be able to breathe as well because there's going to be constriction of those bronchioles. And one thing I love to talk about here when we talk about allergic reactions is the way to stop an allergic reaction. So let's just think about those two allergic reaction symptoms that we just talked about. Constriction of the breathing tubes and dilation of blood vessels. Can we think of a way that we could reverse an allergic reaction given those two symptoms that we just talked about? Well, we do know of a chemical that has an opposite effect to that allergic reaction. We know of a chemical that can constrict blood vessels and increase blood pressure and dilate our breathing tubes and increase respiration. And those are going to be those things from the sympathetic nervous system that give us our stress responses, like norepinephrine, and even epinephrine can do that. And in fact, when someone's having a severe allergic reaction, we can take a little device, stab it into their muscle, and inject them with epinephrine to reverse the negative effects of an allergic reaction. And we call those things EpiPens. They are pens, or these little needles that can inject epinephrine into our blood and reverse the negative effects that these basophils have during an allergic reaction. Kind of fun and interesting there, right? So those are the granulocytes. Let's next talk about the agranulocytes. I'm just going to kind of skim over these ones. We've got lymphocytes, which are a major player in our immune system. And one thing that they can do is produce antibodies, which are these little proteins that search around for a perfect match of their structure. And if they find that perfect match, they glob around this particle that's in our blood, and it kind of triggers an immune response when we try to attack that thing. So antibodies, one of the major things that lymphocytes can produce. I'm not going to ask us too much about this, but lymphocytes come in a couple different forms, like B cells, T cells, and natural killer cells that we'll talk about a lot more in A&P2. So I'd just like us to be like a little bit aware of these things. Next up, the last white blood cell that I'd like us to know about are these things called monocytes. And monocytes are the precursors to macrophages. So they're kind of floating around through the bloodstream, and they're looking for like pathogens potentially, but really like dead cells, debris, and they play a major role in the immune system. And a nice way that you can think about it is that neutrophils come in at the initial stages of an infection or like foreign bodies making it into the bloodstream. And then monocytes come in afterwards and clean up the battlefield. They're sort of like engulfing and cleaning up all of the dead neutrophils and dead pathogens that might have died on that battlefield. Given that we have all of these different kinds of white blood cells, and we have red blood cells and platelets in our blood, we have an interesting way of generating all of these different cell types. In our red bone marrow, we have these things called hemocytoblasts. And hemocytoblasts are stem cells that can give rise to all of the formed elements that we just talked about. We can, if we need to create more red blood cells through erythropoiesis, these stem cells can divide and turn into red blood cells. If we notice that an infection is taking place, we might want to create a bunch of neutrophils or a bunch of lymphocytes. Or they can also create the last major part of the formed element that I'd like us to talk about, which are platelets. So let's talk about that one next. Platelets. What do platelets do? Well, platelets are just these like little fragments or little particles that are kind of shedded off of those stem cells, the hemocytoblasts. And platelets float around through the blood and hang out, and a lot of times they just sort of float through the blood and have no function at all. But what they're looking for is they're looking for damage to blood vessels. And their job is to patch up and prevent blood from flowing out of damaged blood vessels or cuts in the blood vessels. And what they do is, when we get damage to a red blood cell, we can go through this blood clotting process. And so I'd like us to know this blood clotting process. When our blood vessels get damaged, a cut occurs in a blood vessel, the first thing that happens is something called a vascular spasm. A vascular spasm. And what this is it's the smooth muscle that's wrapped around the blood vessel. When it gets damaged or cut, it has this reflex to contract and prevent blood from moving through that blood vessel. Sometimes it doesn't prevent all of the blood from moving through it, but it restricts and reduces the amount of blood that can move through it. Then, when the blood vessel is damaged, the platelets that are on the inside of the blood vessel now can see the connective tissue on the outside of the blood vessel. Normally they can't see it, but if there's damage to a blood vessel, the platelet then notices that it can see the outside of the blood vessel, and it gets really excited. It's like, okay, it's my time to shine here. I'm going to activate and do my main job, which is to become very sticky, release a bunch of clotting factors that activate more platelets and attract a bunch of platelets to that area. And what they do is they form something called a platelet plug, which is these platelets get very sticky, they release a bunch of clotting factor, they attract more platelets to the area, and they all stick together and try to block up most of the blood flow through that blood vessel. If it's a really small blood vessel, sometimes they can completely block all blood flow through the area. Other times they block like a lot of the blood flow or most of the blood flow in that area. So they get really sticky, they form a platelet plug that starts blocking the amount of blood flowing through the area, and then they're releasing a bunch of this stuff called clotting factors. And what they do is they create a chain reaction of chemical conversions, and I'd like us to know this reaction here. The clotting factors that are released from the platelets that are forming the platelet plug, they first interact with a chemical that's floating around through the bloodstream called prothrombin, which is this inactive chemical that's just kind of floating around, kind of ready for any cut that might occur in the body. And when these clotting factors see prothrombin, it converts prothrombin into thrombin, the active form. And thrombin then finds another inactive chemical called fibrinogen. And thrombin converts fibrinogen into fibrin. And the way you can think about this fibrinogen-fibrin conversion is fibrinogen are like these really slippery noodles floating through the blood. And when thrombin comes up to fibrinogen, it converts that slippery noodle into a really, really sticky noodle, like a dry noodle, and they're all going to stick together. And this fibrin, the active form, forms a mesh that will all stick together, and it'll grab onto the platelet plug, and it'll hold that platelet plug in place, which will prevent most of the blood flow from moving through that area. And it sort of holds on to the platelet plug, it holds on to the sides of the blood vessel, and it kind of blocks up blood flow through that area. Here's kind of interesting part. Once these platelets form this platelet plug and form the fibrin mesh, the platelets start to shrink, and the fibrin mesh pulls on the blood vessel, and it pulls the open parts of the blood vessel together, closing off the cut and allowing those cells to quickly heal and seal up the damaged blood vessel. How cool is that? The platelets start shrinking, and they pull this mesh together and seal up the blood vessel for the endothelium, epithelial cells to heal back up, and then the connective tissue on the outside to grow back together. Once that process has taken place, a second process comes in called fibrinolysis, which is where these fibrin mesh tubes get cut up, and then the clot can just freely flow away. So that's sort of the basics to the clotting mechanism in the blood. And the last thing I'd like us to talk about is blood typing. Let's talk about blood typing next. You probably have heard about blood typing. You might have even know your own blood type, like I'm O positive. Let's talk about human blood typing. Humans have 3 major types of blood: A blood, B blood, and O blood. And then there's this second categorizing way that we're also going to talk about called an Rh factor, which you can either be positive or negative for. But let's think about Human blood. Humans really do not like to get someone else's blood in their own blood. It can be really bad for a human to get a different person's blood in their own blood. It can have different pathogens in it that can attack human's blood. It can have diseases in it. It's not ideal for human blood to mix with other human blood. So our body has formed a defense mechanism where if we notice that we have someone else's blood in our body, we have the potential to attack that blood, clot it up, break it down, and eliminate it right away. And this will happen even with like other tissue that can be transplanted into humans, but we're going to stick to just talking about blood. And so the major different types of blood, again, that we can have is A blood, B blood, and O blood, and each of those can either be positive or negative. And here's what each of those means. On the surface of red blood cells, there are these little markers that tell the body what kind of blood it is. And these little surface markers are called antigens. Antigens. And then in the plasma floating around the red blood cells are these proteins called antibodies. Antibodies. And we talked about this just briefly, but what antibodies do is antibodies look for foreign antigens, and they want to attack the foreign antigen and sort of clot it up, all bind to it and prevent it from going anywhere. And then your body will realize that and break it down. So antibodies, the way I think about them is they're like security guards that have a list that they're looking for specific foreign invaders, and they'll go up to each cell and they'll say, do you have the markers that I'm looking for? No, you're good. Do you have the marker that I'm looking for? No, you're fine. And if they find the marker that's on their list, they will grab onto it, hold it down, and prevent it from going anywhere, and will eventually break it down. So again, antigens are on the surface of red blood cells, and they determine what kind of blood it is. They're like these little markers. Or maybe an ID that the blood hangs onto, the red blood cell hangs onto. Whereas antibodies are these little security guards that are floating around in the plasma, and they're looking for a specific type of blood, and if they find the type of blood that they are coded for, they will attack it. And that's a really key thing to note here. The antibodies, if they see the antigen that matches themselves, matches what they're looking for, they will attack it and try to eliminate it. So let's go through each of the blood types now. A blood has A antigens on the surface of the red blood cell. A blood meaning they have A antigens, or these little surface markers that denote it as A blood. And by the way, these are just like little shapes on the surface of the blood, and then we give them their letter that we would like. So it's just this little surface marker, this little ID that the blood carries around, and we called it A antigens on the surface of that blood. And in the plasma of A blood, we have the potential to create anti-B antibodies, meaning that if those antibodies see B blood, they will attack it and try to mark it for destruction. Let's keep going here. B blood would have B antigens on its surface, denoting it as B blood. And it would also have, in the plasma floating around in the blood, they would have anti-A antibodies that are looking for A blood, and if they see it, they're going to attack it. AB blood has both A and B antigens on the surface of the blood, meaning that it does not have anti-A antibodies, or else it would attack itself, and it also doesn't have anti-B antibodies on it in the plasma, because it would also attack itself. So AB blood has A and B antigens on its surface and no antibodies in its plasma, because it's got both markers, and if it produced any antibodies, it would just attack itself. O blood, like the kind of blood that I have, it doesn't have any surface markers on the outside of it. It's O, meaning like if you looked at it, it would just like a little O. No antigens on its surface. And then in the plasma, it can produce anti-A antibodies and anti-B antibodies, meaning that if A blood is present, it will get attacked. If B blood is present, it will get attacked because it has both of those antibodies. And in fact, let's just do each one of these together. Hopefully maybe you have a chart or you can think about these as we chat about them. Let's think about what would happen if A blood it's got those A antigens on its surface and B antibodies in the plasma. What would happen if we gave a transplant to that person, if we gave them A blood? Well, the A blood would have A antigens on its surface, and it would be totally fine. The anti-B antibodies wouldn't attack it. It would be totally fine. If we give a person with A blood a B infusion, we infuse B blood with them, give them a transfusion of B blood. The B blood has these little surface markers on it that would get attacked by the anti-B antibodies in the plasma. Same thing with AB blood. If we give a person with A blood a transfusion of AB blood, it would get attacked by the anti-B antibodies in the plasma because it has these surface markers on it that match the anti-B and the antigens on the surface. If we give A blood a transfusion of O blood, the O blood does not have any antigens on the surface, meaning that the O blood would not be attacked by the antibodies present in the A blood. So if you have A blood, you're allowed to get a transfusion from A, and you're also allowed to get a transfusion from O. Let's keep going here. What about B blood? With B blood, you're allowed to get a transfusion with B blood. That would be totally fine. You can't get a transfusion from A because it'll get attacked. You can't get a transfusion from AB because it'll get attacked. But you can also get a transfusion of O blood as well. Let's do AB blood. If we have AB blood with A and B antigens on the surface, we don't produce any antibodies in the plasma, meaning you could get a transfusion from A blood, B blood, AB blood, and O blood. That would be totally fine. And last but not least, if we have O blood, we could not get a transfusion from A because it's got those A antigens on the surface that our antibodies in the plasma would get attacked. B could not be given, O blood could not be given a transfusion of B blood. It would get attacked. O blood cannot get a transfusion from AB blood. It would also be attacked. And so if you have O blood, you can only get transfusions from O blood. So those are the basic A, B, AB, and O blood types. But there's also a secondary classification that's important here. There's also a presence of a different receptor called a Rhesus factor or an RH factor. And each one of these blood types can either have the antigen for RH on it or not have the antigen for RH on the surface. So each of these blood types can be positive, meaning it has the antigen on its surface, or it can be negative, meaning it does not have the antigen on its surface. And if we have a positive blood type, meaning it has the RH factor, the antigen on the surface of the blood, it will not produce the antibody that will attack RH antigens. Meaning that you could get either positive or negative blood as a transfusion. But if you are negative for the RH factor, you are not producing the antigen on the surface of the blood, meaning that there's a potential that you could produce the antibody that would attack positive blood. And so if you have negative blood, you shouldn't receive a transfusion from positive blood, but you can receive a transfusion from negative blood. So we'll go through each one. If we have A positive blood, you could receive blood from A, and you could receive blood from O, and you could receive either positive or negative. If you have A negative blood, then you can only receive A negative blood or O negative blood. Same thing with B. If you have B positive, you could receive blood from a B donor or from an O donor. And if you're positive, you could receive B positive or B negative, O positive or O negative. But if you have B negative blood, you can only receive B negative or O negative. With AB, you can receive A, B, AB, or O blood types. And if you're positive, then you could receive the positive or negative of any of those things. So AB positive can receive any kind of blood because it doesn't have any of the antibodies in the plasma. But if you're AB negative, then you could only receive A negative, B negative, AB negative, or O negative. You can only receive the negative type. And interestingly, if you have O blood types, remember you can only receive O blood there. If you have O positive, you can receive O positive or O negative. But if you have O negative blood, you can only receive blood from O negative. But what's interesting here is that O negative blood can actually be given to any of those blood types. If you notice, whenever we talked about them, O negative was present in all of the blood types that we could give a transfusion to. So O negative blood is super important for us to have on hand, because if we don't know someone's blood type, we can just automatically give them O negative blood because it's going to be accepted by any blood type. So if you have O negative blood, I'm sure like blood banks call you all the time and they're like, hey, can you come in and donate some blood? Because it's really nice to have a bunch of that stored up. The only maybe little caveats to this, I'm not really going to ask us about this, is that if you have O negative blood, sometimes you don't actually make the antibody for the Rh factor until you're exposed to positive blood types. And the main way you get exposed to positive blood types is usually through pregnancy. Either mother or the fetus has the opposite blood type, and you'll get exposed to the positive blood type and then start producing the antibody. So a lot of times if you're a pregnant mother and you have negative blood, you'll need to continually get a shot that will suppress the antibodies that are created to attack the positive blood type. Because, for example, if your fetus has positive blood, then the antibodies that are in the mother's negative blood will attack the fetus, and it can cause lots of pregnancy complications, including miscarriages. Any other interesting caveats to this? Sometimes if you have a woman who's potentially could become pregnant, they might just, and needs a blood transfusion, they might just give them O negative blood automatically, even if they have like a positive blood type and could receive positive blood, just because they don't want to expose that person to the antigen if they don't have to. What else? There's also a few other really minor blood types, like a K blood type, but they're kind of so rare that most people don't even really consider them because they don't get exposed to them enough to produce the antibodies in the plasma. So not one that really comes up too much, but you might have heard someone like, it probably only happens like many decades ago, but if somebody gets a blood transfusion from a K positive blood, they might start producing an antibody to fight it off, and so that we will need to be really careful about that one. And I think there's maybe a couple more, like a D blood type. Don't quote me on that last one. But those are the basics of blood typing. I think I would like us to know on each of those blood types what antigens are on the surface of the blood and what antibodies are present in the plasma of that blood, and then what blood types can receive transfusions from which other ones. So good luck studying, and I will see us all soon.